Good morning, everybody. Happy Friday. It's good. It's good that it's Friday. Huh? We're all ready for a weekend. I'm sure I'm ready for a weekend. Oh, goodness. Okay. Any questions before we get started this morning? Yes.
What day are the grading presentations due?
Before you leave for Thanksgiving.
Just
Yeah, I've left the assignment open until next Wednesday. So just haven't done before then. And everybody should be assigned your peer evaluations now. And what I did is I assigned, each of you one peer evaluation for a person that is in the groups that you're not in. And so you should just have two of them to do. And then the results of all of that will get shared. And then once you guys get all that done, I will load all that into a spreadsheet and do my grading thing.
it's for a person in the group.
Yeah, but it'll apply to the whole group.
Okay.
Because it's group project. Yeah. So. I didn't think, I mean, you wouldn't want to go through and have to fill it out like 5 times for the same thing. So, and I used that strategy last year and it worked fine. So. Anything else? All righty, well, let's do it then. All righty, so we were talking about osmoregulation and osmoregulators last time, and I want to start off with just an illustration of how osmoregulation can occur in some organisms that we would consider to be fairly simple. So here's an older study that was done on a particular type of a bivalve, a little clam type creature. They call it a mussel, but it looks more like a clam than a mussel, I think. But key thing here is that in the experiment they did, it was a relatively simple experiment where they took these mussels and they introduced them to a variety of solutions that varied in salt concentration. So here is the basic experimental setup. Here we have basically a measure of body weight on the y-axis here. We have time on the x-axis here. And then they subjected these mussels to 4 different salt concentrations. A dilute salt concentration here of about 3 parts per thousand. Then 27 parts per thousand, 36 parts per thousand, which basically is seawater, and then 48 parts per thousand here. And what they found is that when they transferred the mussels into these various salt concentrations, they found that the animals were able to actually adjust their internal osmotic concentrations to normal. So they were able to osmoregulate. And so if you look at these lines here, all of these lines up here, they start by gaining, at least the 2 dilute solutions up here, they started by gaining a lot of water, and then they corrected presumably by adjusting their internal salt concentration until they return back to control values. The only one that they were not able to make such an adjustment was the 48 parts per thousand, which is a high salt concentration. It exceeds that of seawater. So we're going to try to warm your brains up really early this morning and ask you the question: why, if these mussels could adjust to all the other concentrations here, and the 36 parts per thousand was actually a control, okay, why couldn't it adjust to this one? Go ahead, Abby.
Is it something to do with exceeding the rate potential that we try to distribution? Do they use study for it?
Yeah, I'm sure they did. While that may deep down, if you're really diving deep down the rabbit hole, may have something to do with it, but I'm looking for something a little more ecological here. Yeah. Huh?
I said never mind.
What?
I was gonna say, is it because of like the threshold value, kind of like the birds that we saw, they have like a specific limit that they can like regulate temperature, and then after that, like they just can't anymore.
Okay, all right, so this clearly exceeds tolerance. Okay. But I guess what I'm really after here is why would this not be included in the tolerance range of this mussel? So these mussels live in the ocean. As you probably guessed by the control. Do you need another hint? Do you think, how often do you think these mussels are going to see a salt concentration that is that high? Oh, did a light bulb just turn on?
A little bit, because mussels don't really move. So aren't they
No.
usually in the same concentration, so they wouldn't need to have a big threshold?
Okay, so do you think that they would ever see something that's more salt, that's substantially more salty than seawater? Probably not. But could they see more dilute concentrations? You're shaking your head, yes, Andrew. Why?
Well, depending on, just different environmental factors, if you have like a bunch of, flowing from a river that's right by or something.
Exactly. Or if you live near an estuary or something like that, where you get big tidal fluxes over the course of the day, it's very possible, and it's highly likely that these mussels do actually see some periods where the salt concentration drops, and so they're adapted to deal with that. But this is something that they would never see. So there's no reason for them to develop any tolerance to it. And it's just an example of how even an organism's physiology is really tuned to the things that they are actually going to experience in whatever environment that they live in. There is nothing to create an adaptation, whether it be physiological or otherwise, to some factor that they will never experience. Okay. All right. Okay, so that's osmoregulation. We'll say a quick bit about osmoconformers, because there are some that exist out there. Starfish, for example, are good osmoconformers. And so if we look at this graph here, again, we have a measure of body weight over here, body weight gain actually over here. which basically corresponds to water uptake. And then percentage seawater right here, and you see you get this nice straight line with starfish. Starfish have always been an enigma to me because, you know, they are like the last invertebrate before you jump to the vertebrates on the taxonomic tree. Yet they are a really simple organism. So that's always been puzzling to me, and a lot of that's based on development. So their positioning anyway. So anyway, that's all I'm going to say about that is that you know, they have no mechanisms for protecting themselves from the external environment. Their biggest protection is to exist in places where the external environment is not going to exceed their tolerances. Here is another example of a response to osmoconformation right here. And this is a study, an old study that involves crabs. And it's a useful study for this conversation because it illustrates that not only is water moving in osmoconformers, but salts are moving as well. Okay. And I want you to think back to, remember we talked about sea urchin eggs and the transport of magnesium or was it calcium? I guess it was, as opposed to sodium, and how that changed the osmotic condition of the egg. Remember that? Anyway, you can kind of think about this the same way. So what they did here is we have a measure of water gain on the x-axis up on top up here. We have blood osmotic concentrations, probably more correctly, hemolymph osmotic concentration down here, and then time here on the x-axis. And so when they put these, and these are crabs, when they put them into a dilute solution, they started to gain mass because they were taking on water. And then simultaneously, Schwab measured the osmotic concentration of the crab's hemolymph and noticed that there was a very, very large decrease in the concentration. Now, this dotted line here is important because by Schwab's calculations, that dotted line would be where the osmotic concentration of the hemolymph would end up if the only thing that was moving was water. But in this dilute solution, the crabs were not only taking up water, but also losing salt to the environment. And so the reason why there was such a big decrease in the concentration was because they were not only diluting their hemolymph with water, but they were losing salt to the environment as well. Indicating that, you know, when you're an osmoconformer, conformer, you know, everything is moving across your body surfaces. Okay. Does that make sense to everybody? All right. We ready to talk about the kidney now? Cool. All right. This is kind of fun though, to learn about how other things accomplish all this stuff. You know, I find that fascinating. All right, the mammalian kidney. It is a key organism or organ in, frankly, all vertebrates, because it is responsible for making sure that you retain the stuff you need to retain in order to be healthy and get rid of the stuff that you need to get rid of so that they don't become toxic. So the mammalian kidney is where urine is produced, and it's produced out here in the outer regions of the kidney out here, and then it gets funneled toward the renal pelvis, which is right here, and then dumped into the ureters, which is going to head to the bladder. Okay. Urine is the product of water and various solutes that have been filtered from the blood. And the blood is filtered. The blood that is being filtered is delivered to the kidney by the renal artery, which you can see right here, and taken away by the renal vein. The kidney is divided into two main parts, the cortex and medulla. The cortex is a rather thin layer where the vast majority of the blood supply is delivered to the kidney, and then the medulla is a larger portion, a lighter colored portion on the inner portion of the kidney. Now, the cortex has a rich blood supply because this is where all filtration in the kidney is going to take place. Okay, the functional unit of the kidney is the nephron, and here's a cartoon diagram of the nephron right here. And all your blood, every time it passes through the systemic system, is going to make its way through the kidney to be cleaned. Okay, and to have stuff removed or stuff retained, depending on what the body's need is. Filtration occurs at the front of the nephron at a structure that's called the glomerular or Bowman's capsule. This little structure right here. You have a ball of capillaries that fits into that capsule, and that is again where filtration is going to take place. Okay, and it's specifically designed to filter just about everything that's in the blood, anything that is smaller than the plasma proteins. Here's a scanning electron micrograph of a glomerulus right here. This one's from a bat, but they're good mammals, and so they're representative of what a typical glomerulus looks like. If we were to take a histological cross-section of the glomerulus, you would know, or pardon me, of the kidney, you would know whether you were in the cortex or in the medulla just simply by looking for whether or not the cross-section included the glomeruli. And you can see in this cross-section right here, we got several of them. And so you know that you're in the cortex right here. So blood is filtered at the glomerulus, and the capillaries that make up the glomerulus are specialized because they have these really big pores that are large enough to allow pretty much all the water, all the small solutes get filtered into the nephron. But the pores are small enough to prevent larger molecules from getting through, and specifically the plasma proteins. Okay, so anything larger than 70 kilodaltons, and if you don't know what a dalton is, it's 1/12 the mass of C12. Anything larger than that will not be filtered in the glomerulus. So if you can read it, because it's kind of small, you can kind of see all the things that do and don't get filtered as the blood passes through the kidney. So water, electrolytes, glucose, amino acids, fatty acids, vitamins, urea, uric acid, and creatinine all get filtered. And that's just a partial list. And then things that don't get filtered, blood cells, which is probably a good thing, plasma proteins, large anions, any sort of a functional protein isn't gonna make it through, and that's going to include things like your hormones and things like that. Yeah.
Do you want us to know specifically what can and can't get passed through the kidney?
Yeah, I think it would be a good idea to have some knowledge of that. It's not a difficult list. All right, so the inner portion of the kidney is termed the medulla. It tends to be lighter in color, largely because it doesn't have the same level of blood delivery to it, all those glomeruli. It's not that it has no circulation to it, because as we shall see a little bit later, there's actually some very important circulation that makes its way down into the medulla, but it's just not as densely packed as it is up in the cortex. Here, the histological cross-section of the medulla. You can see a couple of things here. First, you can see that there are no glomeruli. So if we were taking a cross-section of the kidney and you saw something like this, you know that you would be in the medulla. The other thing to note here is note how all the cross section here all seems to kind of orient down towards this corner, which is where the renal pelvis would be. And this is because the tubular nature of the nephrons are all kind of aligned here, and they're all kind of directing everything down towards that renal pelvis. All right, so what we're going to do now is we're going to take a quick walk through the entire nephron. And then once we've done that, we'll come back and dive a little deeper into each portion of it. So we'll start at the beginning, and that will be at the glomerular capsule. The glomerular capsule, which the glomerulus fits into, is found in the cortex. And this, as we mentioned already, is where filtration takes place. So this is where urine formation begins. Okay, right here. And here's a cross section of it right here. Okay, the glomerular capsule empties into the first part of the nephron, which we call the proximal convoluted tubule. So this portion of the nephron right here. And this directly receives the filtrate from the glomerular capsule. If you take a cross section of the proximal tubule, you'll find that the inner lining is covered with microvilli, forming a brush border. Presumably, if you've ever taken any cell biology or something, you've probably heard that term, brush border. You all familiar with it? What's the purpose of it? Why would that be important in this portion of the nephron? What does a brush border serve to do? Go ahead, Abby.
Well, it can increase surface area as well as move solids across.
Yeah. Increasing the surface area is huge here. Okay, so remember back when we talked about surface to volume ratio, I mentioned that it was going to be really a big player in the kidney, and this is the spot where it is, because this first portion of the nephron is where the vast majority of your reabsorption of important materials is going to occur. And so you want to maximize your surface area in this portion of the nephron. Okay, so most reabsorption is going to occur in the proximal tubule. 2/3 of all the water that you filter will be reabsorbed right away. And I'll give you some numbers in a little bit that will illustrate why you really need to have this happen. Okay, more than half of your sodium is reabsorbed here as well. But they're not the only things. You're reclaiming chloride, you're reclaiming a bunch of bicarbonate as well, phosphate, glucose, amino acids. So, walk right down the list of all the things that are filtered at the glomerulus, and you're going to reclaim a big chunk of those here because you just don't want to get rid of all of them. And so this is the compensation for having to design a system that is really going to filter your entire blood supply. You take everything out, and then you quickly grab back all the stuff you don't want to get rid of. The other interesting thing here is that things are not just reabsorbed, but you do a little secretion here as well. This is the spot where you will also dump extra protons. So the kidney does play some role in pH regulation, and not just here. We probably won't get to it today, but next week we'll talk about how when you get to the end of the nephron, it actually continues to play a role in pH regulation there as well. Questions about this slide? Just a little side note. Adaptations can occur in kidneys, depending on need. And this here is a study that was done by Giovanni Casati many years ago, looking at these honey eaters. And these honey eaters live in two distinctly different environments. So you've got this little wattle bird here that lives in a fairly tropical zone. and then this spiny cheek tongue eater here that lives in an arid zone right here. And so what Giovanni did is he did some histology on the proximal tubule of these two birds. And what he found is this is actually two cells here. You can see the brush border right there. But what he found is these cells were tightly packed together in the proximal tubule of the species that lived in the wet environment. But when he looked at the proximal tubule of the species that lived in the arid environment, they had these big paracellular gaps right here. And the long and the short of it was that these birds that lived in a dry environment, these paracellular gaps made them really effective at reabsorbing water. The reason why this was an important study at the time is because there's a lot of habitat destruction that's going on. This is over in Australia. There's a lot of habitat destruction going on, and what they were finding is that as these wetter areas become more urbanized, that they're warming up in temperature and becoming drier. And so the take-home message from this was if this species was forced to live in a drier environment, they would probably struggle because they don't have the same water reabsorption abilities that the species that's adapted to live in those drier climates do. So this is an example of how these organisms are fine-tuned to their very specific sort of environment. Okay, when we leave the proximal tubule, then we dive down into the loop of Henle. Okay, and the loop of Henle takes the nephron down into the medulla. So the filtrate, what's left of it, is going to move from the proximal tubule down into what we call the descending portion of the loop of Henle, where it will then make a U-turn, head back up the ascending loop, and eventually make its way back to the cortex. And so you can see that right here. So here's the descending arm of the loop, and here's the ascending arm of the loop here. Interestingly, it is generally believed that the ability of an organism to concentrate its urine. So you remember last time we talked about how we don't have the ability to concentrate urine sufficiently to drink seawater. Seawater is 3.5% salt. We can't get our urine salt concentrations up higher than 2%. And that is believed by many to be related to the length of these loops of Henle. So the longer the loop of Henle, because of the way that they work, and we'll look at the details of those not today, but next week, the longer the loop, the more concentrated your urine can become because of what goes on in the loop. Okay. And this all has to do with a couple of countercurrent mechanisms that occur with the loop of Henle. And just as a little highlight teaser, when we get and talk about the details of loop of Henle, we're going to talk about the difference between a countercurrent multiplier and countercurrent exchange. They are not the same thing. So I want you to get your brains trained to separate those two concepts when we get to it. Both do occur in association with loop of Henle. Now, you may or may not have known that in your kidney, you actually have two different types of nephrons. You have a simpler, what we call a reptilian type or cortical nephron that has a very short loop of Henle, illustrated right here. And the loop just barely dips down into the medulla. We don't hear about those very much. And then we have the more standard, more familiar type of nephron that we refer to as a mammalian type or a juxtamedullary nephron. And these are the ones that have the longer loops that dip way down into the medulla. And it's these juxtamedullary nephrons that are the big players in concentrating urine. So another way to evaluate the urine concentrating ability of an organism is to look at the ratio of these two types of nephrons in their kidney. All right, so urine concentrating ability has been evaluated for animals in general, but mammals several times. But one of the more recent studies to show you that once we got past a certain point, we didn't do it anymore. Carol Bashaw in 1990 did a fairly extensive study on this, where she correlated urine concentrating ability with the thickness of the kidney medulla, and using the thickness of the medulla as an index for how long the loops of Henle are. Does that make sense why she would do that? So here are the key data from her study, and we're going to start with the graph on your right over here. So we basically have urine concentration on the y-axis here, medullary thickness on the x-axis here. You can see you get a nice straight linear correlation right here. And it turns out that this relationship has an R-squared value of 0.59. What does that tell you? How many of you know what an R-squared is? Give me.
I don't know if I can give you an explanation. But I
Give it a
remember
go.
it from stats.
Can you give it a go?
Yes, something to do with the line of strength, isn't it?
Yes, it does. So what R-squared tells you is it tells you what proportion of the variation in your dependent variable, which would be your y variable here, is due to variation in your independent variable, your x variable here. So in other words, how much of the variation in urine concentration is due to variation in the thickness of the medulla? And so what this relationship, this simple model would tell you is that medullary thickness explains 59% of the variation in urine concentrating ability. What's your response to that? Is that good? You're all skilled scientists. We could even round it to 60%. We won't split hairs for 1 percentage point there. No opinions? Zach, do you think this is a good number? He said with great confidence. Susanna, is it a good number?
I don't know.
You have no idea?
I don't
That's an
remember.
honest answer, though.
Yeah.
Anita, what do you think?
Don't look at me, I don't know.
I'm looking at everybody. I can look at Emmy above the nose. What do you think?
What was your question again?
It is so Friday, isn't it? Is this, would you consider this I'm gonna reword the question. Maybe it'll help you to answer it. Do you consider this relationship right here with an R squared of 0.59 to be good evidence that the length of the loop of Henle is a major player in urine concentrating ability? How's that for a rewording of the question?
Yeah, I mean, I think if that's the question, then it's pretty clear that this has a major effect, you could say, but it's not the only thing that has an effect. So there's other factors as well.
And that is a fair answer, because while it explains 59% of the variation in urine concentrating ability, it does not explain 41% of variation. So there are other things that that will impact how thick that medulla is. All right. And so we look at the graph here on the left. So here we have the thickness of the medulla on the y-axis now, and body mass down here. And you can see that body mass alone explains 46% of the size of the thickness of the medulla. So the take-home message from this is it probably does play a role, but it's not the only thing that that is playing a role in how thick the medulla is. Okay. Now remember, the thickness of the medulla is being used as an index for the length of the loop of Henle, and that would be yet another question, is that whether or not that is the best way to measure that or estimate that. It certainly is an easy way to measure it. Okay. So we've gone down, done our U-turn in the loop of Henle. We have come back up, and now we enter the distal convoluted tubules and collecting ducts. And the reason why we talk about the distal convoluted tubules and the collecting ducts together is because they really function together to do what they do. You really can't separate them. Even though the collecting ducts technically are not part of a single nephron, you have a bunch of nephrons that will join into a single collecting duct. But the collecting duct and the distal convoluted tubule actually share a role. Okay. The one thing that you'll notice about the distal convoluted tubules that are different from the proximal tubules is you don't see a brush border here. And the probable reason for this is that by the time we get to this point, whereas we're doing bulk transport of stuff we want to keep in both the proximal tubule and the loop of Henle, but when we get to the distal tubule and the collecting ducts, now we're in fine-tuning mode. Okay. So we are going to fine-tune the regulation of a bunch of ions. We're going to fine-tune how much water we're going to reabsorb. And do I have it on here? Yes. This is the one region, or these are the regions of the nephron that are under hormonal control. The proximal tubule is not under hormonal control. The loop of Henle is not under hormonal control, but portions of the distal tubule and the collecting ducts are under hormonal control. So, for example, we've talked about antidiuretic hormone multiple times. Okay, this is the region of the nephron where antidiuretic hormone is going to act, vasopressin. Okay, we mentioned aldosterone. Okay, this is the region of the nephron / collecting duct where aldosterone is going to act. Okay, so here, this is just a table, and we don't need to take a lot of time to walk through this because it just repeats things that we said. But this is a table in the Widmeyer text that just kind of summarizes what each of the regions of the nephron does. So, you know, maybe you'll find that useful. All righty. Let's go back to the start and dive a little deeper. So at the glomerular capsule and the glomerulus, we form an ultrafiltrate, okay? And the filtration, as you might guess, is driven by hydrostatic pressure. So you remember net driving force, net filtration pressure? Yet another place where this plays a role. The net filtration pressure is going to favor the net water movement out of the capillaries and into the capsule. And you can actually see some pressure numbers down here for this. So you can see that the pressure inside the capillaries is 6.7 kilopascals. And then the capsule pressure, you've got a colloidal osmotic pressure of minus 3.5 kilopascals. With hydrostatic pressure of minus 1.9 kilopascals, the net pressure difference is 1.3 kilopascals, going in the direction of the big arrow right here. Now, something that's really interesting about this is it doesn't matter what vertebrate you are looking at. It doesn't matter what the blood pressure of the vertebrate you are looking at, the net filtration pressure in the kidneys at the glomerulus is always going to be right around this 1.3 kilopascals. So there seems to be something magic about that number. All right, so you filter at the glomerulus. You're pumping most of the water out of there. You're pumping most of the small solutes out of there. You're leaving the proteins behind. And those proteins do have an impact on the osmotic pressure. So the osmotic pressure inside the capillaries is going to go up. And so keep that in mind because that will become really important once we get to the loop of Henle and start talking about how all that works. Well, actually, it's even important for here because when we get to the proximal tubules, we are reclaiming the vast majority of the water that we have filtered. And that increase in osmotic pressure inside the capillaries is really important for drawing that water back into circulation. Okay, fluid movement, the whole filtration process is driven by the Starling hypothesis, and I'll go ahead and put all that up here right now. Basically, this is just a repeat of the net filtration pressure equation. Again, the only addition here is this value for K, which is described as being a fudge factor that is responsible for variations in how fast fluid is flowing. But beyond that, it's the same equation for predicting whether fluid is going to flow into or out of a capillary that we've looked at a couple of other times. But in this particular case, it's referred to as the Starling hypothesis, and the various osmotic forces here and pressure forces are referred to as Starling forces. So. Okay, the rate at which blood is filtered at the glomerulus is called the glomerular filtration rate. In humans, It is about 115 mils per minute for women, a little bit higher in men, 125 mils per minute. The mass specific glomerular filtration rate is about 1.5 mils per kilogram per minute. And tuck that number into your brain for a few moments, because we're going to compare our GFR to those of some other creatures in a moment. We have about 5 liters of blood in our bodies, which means that at this GFR, our blood gets totally filtered every 40 minutes. Pretty fast, huh? Now, here's the reason why it's so important to immediately reclaim all that water that we filter in our kidneys, because filtering at such a rapid rate that we are losing water quickly. So if we don't reabsorb it, we literally would urinate ourselves to death in less than an hour. Okay, just look at GFRs that have been measured in some other organisms. They vary a little bit depending on habitats that they're adapted to. So here's a glaucous winged gull, so a seabird. They have a little higher GFR than humans do. It's about 2.3 mils per kilogram per minute. Budgie, anybody ever have one of these? They are native to Australia, but they're a common pet bird in this country. In their native habitat, they live in very, very wet environments. So in a lot of ways, like we've talked about how hummingbirds, because they're nectarivores, they turn over a high proportion of their body mass in water every day. The GFR is really high in the budgies, almost 4.5 mils per kilogram per minute. And this can pose a real problem for them, because they were at the center of a major dry period, major drought, hot spell in Australia, and where it was not uncommon. These are all budgies that have found a shade underneath, somebody's little shelter, and they just went in there to get out of the sun, but just literally, millions of these things just died because they were not designed to retain water. And it was so hot and so dry that they lost water so quickly, and a lot of them just couldn't survive. Here is an organism that is actually adapted for a drier environment, the Gambel's quail. GFR is a little bit less than that of humans, so about 1.39. Remember old Phylomedusa here, our frog that lives in the canopies of South American tropics. When they are in water, their GFR is very similar to that of humans, but if they're out of water, they can shut it down. They have mechanisms to stop or slow down the functioning of their kidney to retain water. So yet another adaptation for water conservation in this particular organism. Toads. This is a boreal toad. Actually lives in fairly cold climates, but again, GFR in water is about 1. Out of water, they basically shut their kidneys down to conserve water. Okay, GFR is in part regulated by the sympathetic nervous system, and we'll look at how is that a hand up?
Yeah. I was wondering, because GFR, he said that they would shut down their kidneys to conserve water. That means that there's less solids being filtered out of the blood, does that have a negative?
And that's a good question. And my take on that is you would never see something like that in a warm-blooded animal. But in a cold-blooded animal that probably is not producing the same level of waste products, could probably get away with it for certain periods of time. Plus something like the boreal toad is going to live in a really cold climate, which is going to dampen down their metabolic rate even further. So that's probably how they get away with that. And they probably have some sort of sympathetic control that just clamps on arteries servicing the kidneys just to shut blood flow off. And they may even divert it. I don't know. Okay, so anyway, the kidneys are innervated by the sympathetic nervous system to some degree, and that will control blood flow. And it's primarily going to work through vasoconstriction. Birds also can use hormonal means of regulating blood flow. They have a hormone that's called arginine vasotocin, AVT, which is similar to antidiuretic hormone. That for them appears to also impact GFR. So it would be a regulatory hormone for them. And that would, again, anytime you're going to be impacting GFR, it's going to be through vasoconstriction, vasodilation. Those are the mechanisms that's the mechanism that has to be controlled in order to change GFR. When you look at your kidneys, they appear, oh, I'm over time. Okay, I'll finish the slide and then I'll stop. They appear to be very capable of maintaining homeostasis with regard to GFR. So like when you exercise and you get your blood pressure up, the kidneys have to adjust to that so that GFR doesn't get too high. Okay, and so they have what's called some mechanisms that are referred to as renal autoregulation, and we'll talk about those on Monday, that allow them to keep a relatively constant GFR. Okay, and anyway, we'll stop there and we'll talk about renal autoregulation on Monday. Questions?
I was like, waiting to